Welcome, everybody. It gives me great pleasure to introduce to you Dr. Aravinda Chakravarti. If I really went through his CV, which is worth its weight in gold, it'll take very long. So I'm going to summarize his achievements and all that has happened through the years. So he received his schooling in India, but then went on to do his Ph.D. in the University of Texas Health Science Center at Houston, Graduate School of Biomedical Sciences. His postdoctoral training, and I must say that his training is in statistics and mathematics in the beginning, and then human genetics. So he combined the two and did a genetic epidemiology postdoctoral training at the University of Washington in Seattle. So he's had multiple academic appointments, the last of which was the director of the Center of Human Genetics and Genomics, the current position in New York University School of Medicine in New York, and also is a member of the Institute of Systems Genetics in the New York University School of Medicine. I must of course mention that he was in the Department of Medicine until early this year. He's just left, but he continues as an adjunct professor in the Departments of Medicine, Pediatrics, Molecular Biology, and Genetics. So he has been a very, very active member of Hopkins all of these years, and he has more than 350 publications. But what I must emphasize is really that he has been one of the pioneers in this field. So we know, we have learned, and we have heard many, many lectures about genes that are responsible for cancer and so on. But when it comes to common diseases like mental illness, cardiovascular disease, etc., we have great difficulty in pinpointing what genes might be responsible so that we are able to target them to reverse these changes. So his laboratory focuses on formulation, development, and the application of genetic, genomic, and computational technologies. This combination of fields is what has made his laboratory and his work very powerful. So they then discover genes and mechanisms in a variety of complex, non-Mendelian human diseases, and they have used, and how do you do this? When we have a gene and we want to study its importance, we just make transgenic, knock out mice, etc. But these are diseases in human, and so what they have to do is to really study families, and they have now two developmental disorders of interest: neuronal function, Hirschsprung's disease or autism, and two late age of onset cardiovascular disorders, hypertension and sudden cardiac death. I am very, very pleased to have Dr. Chakravarti come today and give us the history of this area of medicine. Thank you very much.
Thank you very much. Thank you very much, Sarah, for inviting me. And thank you all for showing up. It's a beautiful day outside. Like you, I would rather also be outside. So I have a particular onus in trying to make sure that you don't go away too disappointed. You might go away somewhat disappointed, but let's see what I can do. I think in thinking about first coming to give a talk here, you have to understand that I lived here over 18 years. I have a particular fondness for both the city and this institution. So I can't say that I've left it completely, and you will see through much of the work that we've done how much really Johns Hopkins has, and Johns Hopkins in this case means the faculty and people in this have influenced my work before I came to Hopkins, while I was a student, throughout my early faculty career, while I was here, and still as I've left it. So I think what I'm going to do is really take a very historical view. I have quite a number of slides. I'm not going to go through any of them in any great detail. For those of you who are particularly interested, I can leave them behind and you can probe me about some of the details later, but I'm just going to sketch some broad themes as to what the field, and I have to tell you what the field means, what the field of human genetics really means. So this is entitled Human Sequence Variation and Its Effect on Human Disease. And I can't tell you that we understand it completely, but we are at the very beginning stages since the sequencing of the human genome to at least have an inkling of what might be going on. So I think this is a very long standing philosophical questions that we have always asked ourselves as to whether we are unique as a species in some way. And in some ways we are, but in order to bring us back to the ground, you have to read perhaps the Nobel lecture by Sidney Brenner where he said, we study model systems for various reasons in biology. One is to understand the mechanisms by which genes act. But when it comes to genes affecting disease, the reason why we study humans is perhaps, as he put it, and for those of you who know the personality of Sidney Brenner would know this, he said, because we are model systems for God. You could take that as you will. So I would say, despite my own biases, that human genetics as a field really has many unique parts to it. Today we meaning wide swaths of people in academics and outside, have a particular interest in human genetics as a way of informing on the pathophysiology of disease. And there are many, many of my colleagues here who do this throughout the institution. And we have a long history. By the way, if I lapse into we, I hope you will forget me, meaning forgive me. I no longer get paid by Hopkins, but nevertheless, culturally we. And I think the aim and the goal is that we can do this in some systematic way. The second part is we can do this systematically, but also in a hypothesis-free way, meaning we do not have to assume that there is a specific mechanism in any given disease, say an autoimmune disorder like type 1 diabetes. But it is also important to remember that humans are perhaps not the only species, but really the major species in which biology in general is being studied, because we are an outbred species. We are distributed through every single ecology in the world, unlike any other, perhaps except for bacteria, except any other species. And so we have been subject, we are not only subjects in research, but we are subject to biological forces that we often cannot determine, cannot simulate, and cannot study in model systems. And that is a unique part of genetics of the human, because it unveils and uncovers many aspects of the biology that we have not seen in model systems. And this is particularly true in the studies of human disease, particularly in common disease. So this distinction, however, has led to many contentious debates. Human genetics, as you will understand, is only now becoming experimental in the cells. In the sense that we can study human cells, we can simulate many biological phenomena in model systems, but it is still largely an observational science and can be. That's where we begin. And it's because of this mix that it is not only an experimental science, that it's led to many contentious debates that many, I would say, till today remain unresolved. And I think because of this lack of resolution, we still find it easy to discover genes that are found in a disease. And I think we are still fairly deficient in explaining disease. So to give you a sense of some of the contentious debates, I've only outlined a few. In the early beginning of genetics that I'll point out, there was this Mendelian versus biometrical debate that's about the nature of inheritance, how much of it was single gene, how much of it was multigenic, whether it was discrete or continuous, a debate that we have till today. This was followed by the genetic load controversy that we've all forgotten. But embedded in that was the debate, what is the role of de novo mutations versus the effects of segregation of, say, a mutant from a heterozygote to the next generation? This, of course, was important and was highlighted when in fact the testing of atomic weapons came to the fore, because the concern was that it would raise the human mutation rate. Again, a subject that we have not resolved. There was, of course, the selective versus neutral evolution of the genome. That is, how much of the genome contributes to the difference between us in fitness. After all, we are only passers-by in the Richard Dawkins sense. We are only vessels to carry our genes, to give to the next generation. And in that sense, really what matters is fitness. But in the current literature, there is a very big conflation and the lack of distinguishing of how much of the genome contributes to a phenotype and how much of the genome really contributes to fitness differences. There's, of course, the famous or infamous common disease and common variant hypothesis that many of you struggle with and I struggle with. Well, I struggle no more. I'll tell you why. And then finally, it's the nature of adaptation. We've all been led to believe that single genes, for example, the gene that arose in Northern Europe, but not only in Northern Europe, also in many parts of Africa. Now it looks like it could be in other parts of the globe as well. A gene that allowed some of our fellow human beings to digest lactose, whereas others not to as adults. But the nature of adaptation hardly ever through single gene exemplars such as that. It is also that adaptation occurs across the effects of many, many genes. The question is, why has this field that we always celebrate for its successes, and we should, why did this field in human genetics have so much debate, so many unresolved problems? And I believe that it's mostly because many of the genetic arguments that deal not with only me, but all of you here and entire populations around the globe is essentially a quantitative and a population argument. And I think partly the reason for the debate is most human geneticists are not trained in this field. In fact, most very religiously avoid doing anything very quantitative. We like bioinformatics, we like databases, we like computation, but population genetics is none of those. It really is the articulation of what genes do in ensembles of people and ensembles of genes in a genome. These arguments are mathematical and quantitative, and I think many biologists suspect these arguments because they embedded assumptions. And by the way, we are not alone in the scientific field to have them. There are many untested assumptions in physics still today, in cosmology, and people vociferously debate them. And in a way, I think it is good for the field. The other is that many on the other side, if I could call them that, on the quantitative side, have analyzed data but done very little to probe the genetic biology in some experimental way, to test their theories and hypotheses. And finally, again, in human genetics, often we don't have specific mechanisms. So I'm going to first tell you a little bit on how these ideas or concepts of genetic variation arose, tell you a little bit about why and who the population geneticists were that contributed to this field, and then speak about disease. And Sarah's going to interrupt me as soon as she thinks I'm going over time. So it's very interesting that the first genetic polymorphism was the ABA blood group and was not discovered by a geneticist. And Karl Landsteiner, who started his work in Europe but spent a considerable period of his later life and his discoveries at Rockefeller Institute, then the Rockefeller Institute, began with the observation that he knew that if you took serum from one species and you took red cells from another species, they would agglutinate. And after the discoveries or after the popularization of Darwinian views, species differences were beginning to be appreciated and understood. But Landsteiner did another experiment where he did this with material from different human beings, and he found some would agglutinate and others would not. This was done during 1901 and 1903. His first observation was in 1901, the same year in which 3 botanists rediscovered the Mendelian rules. So he very quickly suggested, he was a very smart physician and scientist, he suggested that these features were inherited, although he didn't do very much about figuring out the inheritance. And he was also the first to suggest that these differences could be used to assess paternity. I didn't know paternity adjudications were a big problem in the early part of the 20th century, but apparently they were. In 1909, with one of his colleagues, he finally demonstrated that the major blood types were AB, AB, and O, which at one time was called C. And this led to blood transfusions using genetic principles to avoid transfusion crises. And the first time this was used in a very, by the way, the first transfusion was something like in 1907, 1908. And they were widely used in World War I. And two of the physicians, Leonid Hirschfeld, he and his partner, his wife, who were working on the Macedonian front, collected blood samples for transfusion purposes on hundreds of thousands of soldiers. And these soldiers on the Macedonian front came from almost every part of the globe. And he very quickly recognized, or the Hirschfelds recognized, that there were wide differences in the frequency of these various alleles. So you could say by 1919, the essential idea of what genetic polymorphisms were, and that they were common, and that they were different between different groups of people, was established. But at the same time, there was another point of view, equally compelling, equally important, that arose from the work of another physician in England, Archibald Garrod. And Garrod was the first to demonstrate that a rare human disorder, it's very interesting as to the story as to how he came upon it, but anyways, he explained that a single disorder, in this case alkaptonuria, had in fact an autosomal recessive basis of inheritance. This is also very early, 1902. And one thing to remember, that all of us who call ourselves geneticists, whether you're a physician or not, is many of us work with data. And of course, every scientist works with data, but Garrod routinely studied families. He went out and collected these families. He collected over 50 such cases and their families. He collected blood, meaning various biological tissues on them, which in this case was urine. And he was also in contact with other experts of the field, geneticists such as William Bateson. And this led him to propose a very powerful and enduring genetic concept. He was the person who coined the term inborn errors of metabolism, but he also postulated that there were differences which he attributed to genes between two individuals, and these differences resulted from missing of false steps in the body's biochemical pathways. And he spoke about this notion of chemical individuality, which of course in today's parlance is what we try to do in personalized medicine. So there were two views, rare mutations leading to a Mendelian phenotype in some families, and these other common variants that existed. And I chose those two examples not in any political way. They were really the first two compelling examples. And by 1910 or by 1920, I think you can imagine, genes were discovered. They were shown to have Mendelian inheritance in man, which was in doubt at that point in time, and both of them proved their utility in medicine. So we don't have to wait very long in the history of genetics to say that genetics could be useful in medicine. But the commonality of many of these alleles bothered many individuals, and in fact, if you read the early literature of genetics, you will find that there were many, many misconceptions of what the consequences of Mendelian inheritance was. Scientists, famous scientists, some of those mentioned here, routinely confused the 3:1 segregation of a recessive, of a dominant recessive that you would see in a single family versus what would happen in a population. And even the great Yule and Greenwood assumed implicitly that all of these alleles had and the reason why they were confused is they assumed that everything had a frequency of 50%. So a remarkable departure from that was studies that were done by Wilhelm Weinberg. He was working alone. He was an obstetrician, and he was also very interested and curious about genetic inheritance in a whole variety of situations. He studied twinning, he studied a number of infectious disease, and he was also studying families. And he wanted to find out what were the expectations of Mendelian inheritance if he considered a pair of siblings. In order to do that, he was smart enough to figure out he had to have had some expectations, which is how much similarity should I expect if these two individuals were random as opposed to related? And that is what led him eventually from the single family laws to the population laws that are now called the Hardy-Weinberg law. Godfrey Hardy, a mathematician, was brought into this fray, and he solved it very simply. But I think personally, as a geneticist, I give Weinberg far more credit because his approach to the problem is the modern way in which we look at the Hardy-Weinberg law, and he was persistent in finding a solution to a problem that existed rather than simply Hardy's mathematical puzzle. So as this happened, the two kind of contrasting examples of Garrod and, say, Landsteiner, what it showed us is that it immediately led to a debate between two schools of geneticists. The Mendelians, here represented by William Bateson, and the other are the biometricians that was represented here by Carl Pearson and his mentor and teacher, Francis Galton. The latter, Pearson and Galton, were interested in the inheritance of metrical traits. In fact, Pearson, Galton first, and then Pearson had invented the concepts of regression and correlation that we use till today. And Bateson, on the other hand, focused on the single qualitative trait differences, usually very rare, through which Mendelian inheritance could be demonstrated in the human. And they disagreed on whether Mendelian inheritance could accommodate both of these kinds of phenotypes. I would say we still have this debate. And I should say that Galton also introduced this nature versus nurture sort of terminology, again, something that is carried on today. But was there really a difference of opinion or debate? I would say probably not, because at about the same time, this is 1919, Ronald Fisher, who also is one of the founding fathers of modern statistics, Fisher demonstrated that these complicated metrical measurements and traits like height, that these multifactorial traits were nothing more than the additive effects of small changes at many, many genes. So he provided a theoretical resolution, but what we often forget is shortly thereafter, in 1920, Hermann Muller and one of his colleagues and friends, Altenburg, they came across a trait called truncate wing in Drosophila. You have to remember the time when the Columbia Fly School was predominant. They were isolating mutations like there was no end of time. And when they came across truncate wing, because it didn't conform to their expectations, they just put it on a side. So of course, science doesn't function well if we only study the things that we can explain. And that's exactly what fly geneticists did. And truncate wing, Hermann Muller, one of many rebels in genetics, worked with Altenburg to do some very elegant breeding experiments on truncate wing, which they call an inconstant and modifiable character, meaning in a cross, they could never control all aspects of it. But nevertheless, they mapped 3 components, 2 on the autosomes and 1 on the sex chromosome. In fact, they further showed that the sex-linked factor was not the sex-determining factor in flies. I think that was a very elegant thing to do at that time. And In going back and looking at these papers, you would say that, in fact, they had shown the essence of what multifactorial inheritance could be. But Fisher's model, and it was a model, was very far-reaching, in fact, was far ahead of its time, because that model required him to postulate that the alleles had very little differences with one another, and that they were common, and that there were many of them, which I think after 80 years we still believe that that's the case, and we believe that that's the correct view. So this forced on geneticists a dilemma that you could not only study genetics in the context of just a single cross, even though you had to explain those observations, but also had to explain the genetic nature of a phenotype in the population at large, and led to the emergence of this subject called population genetics. And even though all the early discussions of population genetics were tied with allelic differences and their impact in evolution, there was still, geneticists were searching for a way to find a cogent explanation for Darwinian thinking. And this led first to a reconciliation, if you will, of Darwin's evolutionary theories, which, by the way, showed that evolution happened, but it was slow, except in certain circumstances, and that these could be explained by quantitative arguments on the distribution of genes in populations. And this led to the development of a mathematical body of work that we now call population genetics that I believe is indispensable. But we still don't teach very much of it when we teach genetics. So this consolidation, I think, was very important, and in fact in the literature has been deemed very important part of understanding biology, and it's been called the evolutionary synthesis. And it's a synthesis because it brought together a body of literature that was normally contributed to by naturalists. It brought paleontological observations together with what we knew at that point in time about the behavior of genes. But the three principal architects that I'll point to who contributed to this field immediately realized that these observations applied equally well to phenotypes, including disease phenotypes. So these were the three main architects. On your left is JBS Haldane. JBS Haldane was an English geneticist, a polymath that contributed to biochemistry, to the origin of life, to many, many aspects of science. He was a popular writer of science. He spent the last years of his life in Calcutta, India, in the institute in which I grew up. And this shows him on the terrace of the building that at least I happen to know very well. And he was recruited there by the gentleman in the middle photograph on the end, who was a physicist called P.C. Mahalanobis, who had this strange collection of academics and scholars in this institute, which is, by the way, which is the only reason why I came to the field of genetics. And sitting between him and his wife, and by the way, he was also, for those of you who are interested, he was a personal secretary for a very famous Indian poet called Rabindranath Tagore, and sitting there in Indian style is Ronald Fisher. And Fisher was also a frequent visitor to the institute, and I showed these particular views of these two individuals simply to say that there was no way that I would not have turned out to be a geneticist. In fact, the puzzle is, why were the remaining students in this institute not geneticists? And the third, very importantly, was Sewell Wright. Sewell Wright really thought of genetics in the context of physiology. And unfortunately, I don't have much time to explain much of his work, which he originally began in the Department of Agriculture, in the US Department of Agriculture, but produced perhaps the most compelling body of work left in population genetics. But what did their theory really tell us about human disease? And I'm not going to read through all of them. The first point is they began to explain why recessive diseases could be common. And they suggested that despite having very deleterious recessive disorders, they're not completely eliminated from any human population, because they arise constantly by mutation. And they are weeded out by natural selection, except natural selection weeds out only the homozygotes, whereas the vast majority of those mutant alleles reside in heterozygotes, which is why the mutant allele has high frequency, even though the incidence of the disease is low, and they persist in populations for a very long period of time, which is why screening is important. Second, dominant disorders also arise by the same process, but dominant disorders have far, far lower frequency for the same degree of deleteriousness because they occur as heterozygotes and they're immediately selected. Third, all of these geneticists, but primarily Haldane, recognized that how do we explain disorders like sickle cell? And that actually he considers thalassemia first. That was widespread in the world in many places and were quite common. And their way of explaining it was to say this was also a question of balance, but here there's balance of losing mutant alleles because of the effect of the disease and losing wild-type alleles because of the effect of some other environmental factor, which in their case, or in this case, was malaria, basically keeping heterozygotes more fit than either. They also demonstrated that the inbreeding effect would be intense for recessive diseases, not dominance. They also showed that the inbreeding effect would be inversely proportional to the frequency of the disease, all of which have led to our asking individuals who have a rare recessive disorder, whether their parents are related or not, and this of course affects the distribution of genes in families and leads to specific genetic counseling. And the last point here of the many that they spoke about is the fact that if you look at X-linked diseases, about a third of all cases every generation are de novo mutation. And this again has many implications in the diagnosis of disease. But despite all of this progress, which of course was actively learned by human geneticists of that era, there was somebody who came up who in fact created a lot of trouble for the field. And that was in fact the person on my left and your left, that is Motoki Kimura. Kimura was a Japanese geneticist who came to work with the gentleman on the right, who was Jim Crow. This is a photograph taken at to the geeks, a famous meeting at Cold Spring Harbor. But Kimura first worked on the classical theory of population genetics, and he extended it. He extended it in many ways in highly mathematical theories using, at that point in time, the new stochastic processes. And after he did it, he struggled with the question of explaining that the so-called evolutionary synthesis, and there's an I should have edited it better, the evolutionary synthesis was not completely tenable. Because under the evolutionary synthesis, you couldn't have as much genetic variation in populations as you saw. The classic way in which this was explained is that, well, if there are two types, one must be less fit than the other, or if it is the most fit in a heterozygote, then both of the homozygotes must be less fit. Less fit is a very elegant way of saying that you need a selective death. That means people are dying because of carrying those alleles. And Kimura argued that, actually Haldane had mentioned this before, that you couldn't carry this on for very long because the same thing was happening at many, many genes throughout the genome. These people knew of the genome. And such an amount of selection was untenable given the fertility that most natural populations had. So Kimura came up with an alternative explanation, and he said that, well, most of the genetic variation we see is neither good nor bad. It's selectively neutral. And because it's selectively neutral, it follows different rules. It's a stochastic walk through the human population as to how long a particular allele persists. This idea was vigorously opposed. I did my graduate studies in one of Kimura's contemporaries, and we were derided for being, and we were called neutralists. These neutralists, very much like where, we red bait and we call people communists nowadays. So So the neutral theory has clearly persisted, and the confusion rose because every variation does not have to have an impact on either a phenotype or fitness. But we need to explain fitness differences if these alleles are to survive. So how do we interpret or carry these ideas forward, or how have they been carried forward in human disease? So one can't come to Hopkins and speak about genetics without really the outsized role of a single person, and that's Victor McKusick. You can see Victor McKusick. Most of you, or many of you, should recognize this is an old version of Herd Hall. And this is where the Department of Medicine has grand rounds. This shows a grand rounds from 1946. And the arrow points to Victor McKusick. He sat on the third row next to Ann McKusick on the right. I actually had a few other photographs from other points in time where he always sat at that position, and he probably sat at that position for 50 years. But anyways, Victor McKusick clearly had learned many of these principles that led to him to study human phenotypes, classify them by inheritance, and when it came to understanding the genetic basis of disease, go out to other populations, such as isolated populations, to uncover the genetics of otherwise rare recessive diseases. Victor clearly established medical human genetics or clinical genetics in this country, but he wasn't alone. Jim Neal in Michigan and Arno Matulsky in Seattle did the same. And for those of you who knew them, they were all remarkable people, and the field that they set forward was very much in their image and their thinking and what they had learned. But among them, I would say, Victor is particularly special. This is Victor at the Bar Harbor Short Course that he began in 1960 that next year we will celebrate the 60th anniversary of. This is Victor pointing to something that was his passion throughout his life, that is the position of a particular gene. He's actually pointing to something that, a gene that I like, which is red cell acid phosphatase on the tip of chromosome 2, that Victor was preoccupied with the idea of creating such a map of the human genome, because that's how he thought we would eventually get to the basis of understanding different genetic disorders. Now this, of course, happened in fits and starts, because most of the field had tools such as broad cytogenetics with not a very great resolution, protein polymorph blood groups like the ABO and rhesus factor and others, and a number of protein polymorphisms that really arose more by chance than anything else. That is, the observations arose by chance. And so it is with particular importance I point out this paper. This is Y.W. Kahn, who in 1978 published this paper, and where he showed that he could use the then new methods of recombinant DNA technology and southern blotting to find a site in the human genome that sat outside a gene that could be used to study the behavior of a gene. He did it to study sickle cell anemia, to show that the sickle mutation, in fact, was associated with a particular fragment. And I'm not going to go into the details, but this could be used in a variety of ways. Because this factor or this site was very close to the beta globin gene, it could be used for prenatal diagnosis in families for serious globin-related disorders. It could be used for tracing the origins of a common mutation like sickle cell. And it really opened the way for using DNA polymorphisms broadly as a way of probing the human genome. All of you probably know that at about the same time, David Botstein and colleagues in a broader sense, Ray White, Mark Skolnick, and Ron Davis. By the way, Mark Skolnick was also in a way both famous and infamous. He had this company Myriad that even though Mary Claire King really mapped and did most of the heavy lifting in mapping the gene for hereditary breast cancer and discovered, in a way, BRCA1. Mark's company actually cloned the sequence. But anyways, these 4 individuals gave a proposal on how we could create a human genetic linkage map. And this linkage map would lead to the molecular resolution of many, at least at that point in time, molecular phenotypes. So this is the current chart. So I'm bringing you back from 1980 to today. This is pretty much the data today, and data that are produced by the Centers for Mendelian Genomics. There are many such centers, but one of them is led by Dave Valley here at Hopkins. And so this is the current status. So about 64% of known Mendelian phenotypes have identified genes. Many of these phenotypes are described in the online version of Mendelian Inheritance in Man, started by Victor, and now the work is carried on by Ada Hamoshier. About 18% of human genes have a known Mendelian phenotype. That's that tan and the 3 gray boxes at the bottom. There are about 30% of human genes that are suspected to lead to embryonic lethality. Personally, I think much of the data is soft. But anyways, that's the current estimate, which means we are not going to find phenotypes in the living. And then there's half of the human genes that have no Mendelian phenotypes attached to them. And I have a lively debate with many of my friends, including Dave Valley, as to whether we will fill that gap or whether they are a different kind of biology. And I'll come to it. One of the reasons I think increasingly we can discover genes, often even in a single family, in a single patient, is because now we have these extensive databases. Mass General and the Broad have been very efficient in trying to take sequences from many, many different studies, such as 120-odd thousand exomes in the EXAC, which is an exome aggregation project, and now a genome-wide project that has additional exomes and about 16,000 genomes, so that when you find a variant based on sequencing, you can look at its frequency in the population, whether it's been observed, if it's observed, what its frequency is, and so it gives you a fairly good idea as to whether it's a candidate variant that might be responsible for the disease. The work doesn't stop there, but it's a very good place to begin. But to me, not to diminish any of this work, this of course is the basic work that we can all contribute to. I think the part that was missing was filled by a very important piece of work that began here. And that is a mapping of common variants and common mutations. And so the first studies was done by Haig Kazazian, who's also faculty here, and with whom I collaborated during these years. And Haig, together with, at that time, his fellow Stelianos Antonorakis, published his first paper in PNAS in 1982, showing that if you had different polymorphisms in a local region of the genome, in this case around the same beta-globin gene, then those sites were not completely randomized with respect to one another. In the genome, if 2 things in the genome are not random with respect to their occurrence, it suggests that they have a common history. That's what that means. So this work led them eventually to find a way, when cloning was extremely difficult and sequencing was painful, that they could take patients with a whole variety of beta thalassemia disorders from around the world in different populations, and they created a consistent, systematic way, an algorithm, if you will, where they could use these genetic markers to say these markers are different from those, or these mutations are different from those mutations, and eventually found the mutational basis, which they did with Stu Orkin. To me, this is a very extremely important piece of work that in my own particular history was very important, because at the same point in time with them, this non-random association that we could demonstrate could be explained by at least a relatively simple genetic feature, which is that there must be recombination hotspots that take these segments in one particular part of the genome and randomizes them with another part of the genome. These kind of associations were some reflection of the physical distance between these markers. And because of that reason, it occurred to me that we could use this as a ruler of time, and therefore we could use it to map associations and map and find genes. I think it's going to be very obscene of me to say that, but I will, which is that other people have claimed that they are the discoverers of this idea, but this work clearly arose out of work that happened here in Hopkins in the early 1980s. And the first test that we could use this in a broad way, not in an isolated population, but in you and me, in any population, in any part of the world, came from this work in which I collaborated with Lak-Chi Choi in the cloning of the gene for cystic fibrosis, or in the molecular cloning of CFTR, and in identifying its most common mutation. So this required making that map at the bottom, which was very, very painful in those days. It was a high-resolution map for those days of genetic markers. And what you see on the cover of Science, which published this work, is this big block of red and other colors, each of them representing a haplotype that was identified in a patient. And by studying these non-random associations, we could delimit as to where the gene for cystic fibrosis and its mutation should have occurred. And the smallest interval also suggested that that's where the mutation would occur, and the different color shows that the common mutation must be that on that red background that turned out to be the delta F508 common CF mutation in Europeans. So this told me that there was a way now, based on all of this history, where we could make these maps across the genome. Genomics was proceeding fast. We were going to sequence the genome, and yes, it was done in draft form in 2000. And so that gave the idea to a number of us that what we could do is make these markers in a much more high throughput way and figure out the associations. And we could use this for not only mapping CF, by the way, it worked for CF because the CF mutation was known to be quite common because the disorder is so common. It had a frequency of about 2%, allele frequency of 2%. But that what we could do is do this as a much bigger project, and that's what led to the HapMap project. And when the sequence of the human genome was finished and modern short-read sequencing technologies such as from Illumina came to the fore, this could be done with far greater efficiency, and this is what led to the 1,000 Genomes Project and others. So this history sort of brought us back and clearly brought me back, and I see Don Arking here, and I have no idea why he joined my lab as a postdoctoral fellow, but I think if it wasn't anything, it was clearly a very exciting time because we finally had the technology that we were deeply involved in at that point in time with Affymetrix. We had these maps that we were creating. We needed phenotypes that we could use for mapping, and the first one that Don worked on was on the QT interval, but this is a map many of you have seen that pinpoints the position of different genetic markers that are associated with various common diseases. It doesn't matter what the phenotype is. But common variants have also been used for many, many other purposes, and they've been popularized. For example, they've been used to trace human history. We are now fairly sure, or we are quite sure, that there was at least one movement, or I should step back and say that modern humans, and I can define what I mean by modern humans later, that modern humans arose in Africa about 150,000 years ago. They resided in Africa for a considerable period of time, about 100,000 years ago, and about 50, 60,000 years ago, we emerged, and this is this out of Africa scenario, and populated various parts of the world, both into Europe, into Asia. There's a southern route that led to the founding of Australia and of lineages in Australia, and then a far eastern route that led to the founding of the New World. So the dates are fairly well known now, not only from genetic studies, but also by connected to other archaeological and evidence and artifacts. And we also know that populations in distant parts of the world, such as in the New World, were derived from ancestors that were themselves derived from another part of the world. So all of humanity now is a series of serial migrations. And these serial migrations also led to mixing with local populations. We had assumed that human beings were unique, but it's quite clear from our genomes that we have mixed in the past with at least two kinds of hominids, and now with data not only Neanderthals and Denisovans, but probably other hominids as well. So nowadays, with Ancestry.com and 23andMe, two companies I really like to dislike, so they have common tests where they use these variants to assess your ancestry. I show this simply because of the story. I'm sometimes pushed into a corner to provide an answer. And this came from this young woman on Gazelle, who is on the side, and she's an NPR reporter, who called me to say, you know, I went and did these tests, and I want you to come and tell me that there are things here that I don't understand. And, you know, I said, okay. And she brought with her her mother, who is a historian, and they both came and they both showed me their ancestry results from two companies. And the question is, her mother has family records that suggest that her ancestors have been in Italy for a very long period of time. But when Gazelle's DNA was tested, she has very little DNA from that portion of the world. One of the problems with ancestry testing in these companies is, I know that ancestry testing can be done well. The question is, are they doing it well? And they have shown very little evidence that their tests are appropriate. So this is one. You would expect that any child to inherit half the genes from any one parent, and she doesn't. So I don't think Mendel was wrong. I don't think, despite the biometrical or the Mendelian debate figures into this, you can imagine what I believe is going on. So we're now getting into using these common variants for other territories that are also quite contentious. So GWAS has implicated many unlinked common variants, but the majority of the phenotype variation is still unexplained. We have said that if we simply study more people, we will fill the gap, but we are not filling the gap very well. And even if we do, the underlying genes are not known and their mechanisms are not known. Nevertheless, there have been in recent time the emergence of something called a genetic risk score, where you take up the effects of every allele that you believe is contributing, and you can come up with a score, and we can take everyone in this room and stratify you and say, with respect to blood pressure, some of you are at the top 1 percentile, some of you are in the middle, some of you are at the bottom, and people are trying to use this for trait and risk prediction. One of the problems is that, going back to the biometrical Mendelian debate, beyond the question of one and many genes, is that these kinds of tests don't separate genetic and environmental factors, which I think if we are serious, we have to do. Showing the effect of genes on a trait is not tantamount to showing that the environment or other factors don't have an effect. I am quite disturbed by the fact, I'm personally quite disturbed, you do not need to be, of an increasing attempt at explaining behavioral and social traits between populations using these kinds of methods. And these include factors that you may think is benign, like educational attainment, or things that, or sexual preference, or things that can be far more contentious, such as IQ. The question is not what we find, the question is what's the meaning of what we find and what we infer we should do next. So in the final few minutes, I know I am running late. I think I'm going to go very fast and tell you where I think much of this is likely to go. So my own lab has been involved in the genetic dissection of some complex disorders. Most of what we've worked on is Hirschsprung disease, which is a disorder associated with the absence of enteric ganglia. It's got many features that make it a fascinating genetic entity to study. It is intensely genetic. And over the last 20 years, I would say that many of our collaborators and I would say competitors, as well as us, have done a whole series of studies to not only find genes that have variants, both common and rare, but as well as try to understand what is basically going on in the disease. And I won't have time to tell you, but basically it's a failure of various kinds of interactions to occur between the gut mesenchyme and these neuroblasts that enter the gut early in development and they need to interact with those cells, proliferate, get cues, get growth cues, such as those that stimulate two receptors. One is a receptor tyrosine kinase RET. The other is a G protein coupled receptor called the endothelin type B receptor, and allows these cells to proliferate, differentiate, and populate the gut. And if you interrupt any one of many processes as part of this interaction, you get a segment of the gut that doesn't have a nerve supply. One of the most fascinating things that is quite some time ago, this is 2005, we clearly demonstrated that a major cause of this disease was in fact not a coding mutation, but rather a very common variant that sits in an enhancer of RET. And this is a variety of data to show that the variant is very common, has a fairly high effect, and is non-coding. And today, from a variety of studies, we know, as it always should be, that in a genetic disorder of any kind, there are many ways to break the biological system. We should not rely on only one kind of variant and have these silly wars about whether variants are common or rare. I show you three classes of variants: enhancers and coding genes, as well as copy number variants. In fact, Joe Tillman, who's a graduate student of mine here, contributed much to this analysis. And I'm not going to go through those details except to tell you that all three categories of sequence variants contribute to this disease, but the enhancers contribute quite a lot simply by virtue of them being common. So we also know genetically, beyond the molecular components and their effects, the totality of the effect, we know that many of the genes, not all, but many of the genes involved in Hirschsprung's disease don't act alone. You can interrupt this pathway or this network, this gene regulatory network, in any one place, and it's going to affect the transcription of many of its related genes. So whether you call this a single gene defect or not may work on the inheritance side, but will not work on the etiology side, because you can interrupt, for example, any one, such as one of those transcription factors, SOX10, and it's still going to affect the downstream regulator, which is the receptor tyrosine kinase RET. So in the next 20 years, there's still many serious questions about genetic variation we haven't solved. One is there are many open questions, as I indicated, on the molecular basis of Mendelian disorders. We don't know that every gene has a Mendelizing phenotype, and we don't know, I'm not convinced that all absent phenotypes at some genes are due to embryonic lethality, and it could be. We don't know, even for Mendelian disorders, how much of the genetic variation is due to coding sequence variants, due to variants in its cis regulators, as well as due to variation in its trans regulators. And this is important because today, even for Mendelian phenotypes, if you do exome sequencing, one can only discover a molecular cause in only about 25 or 30% of cases. So there's much to discover and much to understand. And the last point the last 2 points is, I'm not convinced that all rare entities we see in the human are Mendelian. They're very likely to be much more complicated interactions. And this is important to understand when there are many, many genes that contribute to a phenotype. What is the unifying principle or mechanisms of all of the genes? in Sarah's case, why do all the genes involved in breast cancer, what is it about them that leads them to be associated with breast cancer? I've become particularly interested in synthetic lethality. If a lowly organism like yeast, it's not lowly by the way, we wouldn't have bread, wine, or cheese if you take yeast out of our repertoire, but synthetic lethality is extremely common in yeast. It says you can have a homozygous null-edited gene. It will not show a phenotype unless you homozygous null another gene. And there's no reason for me to suspect that it's not the case in the human. So I guess I'm answering the questions. We are not that unique. Even for complex disorders, the even more open questions. We don't know why is it that we have so many genes for a disorder, and why is Mendelian inheritance of traits so uncommon? We don't understand why the effects are so small. In fact, an intriguing question is, do environmental factors affect the same biology as do genetic factors? In fact, there's a fascinating study. Many of you know the thalidomide story, and it shows that there is a gene, a transcription factor, which when mutant leads to a developmental sort of acuropodia, but thalidomide, in fact, downregulates the same transcription factor. So I don't know that environmental factors can act outside the biology. So finding the biology is important as a way of informing us what goes wrong in the disease. Not that a disease is genetic per se. And again, the question of gene diversity. This, of course, has been put in a very extreme, controversial way, correctly, I would add, by Jonathan Pritchard. This is a paper that really irritates many geneticists to no end, because he has thrown it back in their face to say, if you're finding so many genes, why not every gene? In fact, if you go back and look, that's what the evidence suggests. And I think we are irritated because we don't have an answer. But for most trainees, I think this is a very important view to pursue. I personally believe that we have, like in the small Hirschsprung example, have to go back and look at gene regulatory networks. We've got to find them. I do not mean gene regulatory networks as just co-expression networks or protein interaction networks. There are many other genetic networks. And the reason why I insist on this is we are doing genetic studies on the genome. We are finding sequence variants. So just looking at protein interaction won't do. We have to connect the genome to the transcript, to the protein and functions. And this is sort of a cartoon model of what I believe is one way that we can think, and my laboratory thinks, that the effects of many genes, be they coding or non-coding, affects expression, that affects the activity in one or many ways of a regulatory network, that then alters some cell behavior. And I put question mark because we are completely ignorant of the principles that determine that, and therefore the phenotype. So I'm not going to I think I've said it all. I'm going to thank you for your patience, and I'm glad to answer any questions you have. Please.
I have a very fascinating thought, very fascinating. I have a little bit provocative question. So, don't you think statistics is a little bit too simplistic for such analysis? I worked in the field for the last 10 years, but so the way that, for example, we mentioned Richard Dawkins, the Hopkinsian way of looking at biology is too simplistic to think of.
Too simplistic in what sense?
In the sense that, this is a
You want an
dynamic
atomic level
system happening, interactions happen. So thinking of simple gene is going to do a lot of things.
No, I don't believe that it's, actual biology could be very, very complicated, right? We are not looking at atomic resolution. We are not looking at every water molecule inside a cell. I think the idea in science is to extract a certain level of meaning at some resolution.
Right.
So if I have found every gene in the genome and used it to explain a phenotype and I cannot, that tells me that I need to look at other mechanisms that can explain much more.
Right. So I'm saying statistics is not as powerful. verbal guy you mentioned, the Japanese scientist.
Well, statistics is powerful because we need an objective means to say we are making progress.
Right, right.
So I would say in Joe's humble example in Hirschsprung's disease, we made remarkable progress because we have a ruler by which we can say this is what we knew and this is what we know now. I'm not saying we've made no progress in other complex disorders. That's not what I'm saying. I'm just saying we need to look at the problem in a way that we make much further progress. And the mechanistic details are important.
Right.
So for these multi-phenotype diseases, what you're saying is that for each one of those phenotypes, there is maybe a different pathway that then may also contribute to each other?
I don't know that there's a different pathway. One of the things is we have no idea about how big these networks are. And so in one sense, you can say in a given cell, every gene is connected to everything else. That's the Pritchard omnigenic argument. That very well could be true, but I think for phenotypic explanations, what we need to do is to say, if I perturb this gene, how far does that effect last? So even though if this gene could be connected there, the question is if that effect lasts through only 2 genes, 3 genes, then that's the network we need to extract.
Can you please comment on your work with autism and delta catenin?
I can do that later because that is very specific and I'm just going to use up more time. But basically, I think autism as well as Hirschsprung's disease has a very particular sort of, the problem there is to explain as well as exploit scientifically the sex difference as to what's the meaning of sex difference, which is what that work pointed to.
What do you think, like you mentioned environment a couple of times, and
Yes.
it's really difficult to
Yes.
control for it,
Exactly.
like when we use cell lines, when we use mouse or whatever models. What do you think for a proportion of the genes where we don't have really a link to the function yet could be then explained if we somehow could take the environment into consideration?
So yeah, so that's a very important question. Meaning, I was also vague about what I mean by the environment. I mean, there are many factors that affect our health or our phenotype, even if it's not our health.
And the role of epigenetics.
And the role of epigenetics, which I completely bypassed. But so I would think that one way of thinking about it is how external cues, they could be anything, would affect some proximal behavior of cells. They probably include all of the epigenetic machinery. My bias is they largely would act through the cis and the trans regulatory apparatus. So one way of doing this is you could say that you could take a cell line, and perturb it in many different ways, not by a genetic perturbation, but by some exogenous agent. Change the pH, whatever, change the ionic concentration and such, and see which genes are affected as a result. And I suspect if you're measuring a specific gene or a phenotype, for example, if you were looking at some cell phenotype, then I think my expectation is that it will not be a random set of the cis regulators or epigenetic regulators that would be affected. I think the thing that we haven't been good at is to connect exogenous factors with intracellular factors. By the way, there are some people who've done these experiments. There's a genetic mouse model for scoliosis, and there's at least one group that's shown that simply by changing the oxygen concentration in newborn mice, or not newborn mice, in the pregnant dams, that you can affect the frequency of scoliosis, and they showed that this is what oxygen sensing factor. So exactly how that factor affects that gene, maybe through methylation or some other, regulation is not known. But I think increasingly we will have those kinds of factors. So if you connect, well, I don't have to say, I mean, why only environmental? Age is one of the strongest determinants of the risk of many diseases. Why is it age?
So, thank you very much for that very elegant and informative historical perspective on hereditary disease.
Yeah.
May I please give you a token of appreciation from the Cancer Center for this beautiful talk? And you can take it out.
Okay.
Please come
Okay.
on over here so we can get a little picture. Thank you very much.
Thank you very much. Thanks,